Okay, so we're back to the biotic again. Today I'm going to be discussing the results from 3 studies that evaluate soluble aluminum as mechanisms driving bacterial community structures in soils, ranging from series of scales, from the continental scale to microcosm scale. And I was hoping I'd be the first one to say aluminum today, but beat me to it. And I'd like to savor it because in several years of doing my dissertation work, aluminum is a rather unconventional variable to mention in soil microbial circles. This is a joint collaboration between 4 labs based in the western United States with varying expertise from geology, soil pedology, and microbial ecology. So to orient you today through the series of studies that I have, basically there are 2 objectives with this. And one is to characterize the relationship between aluminum and bacterial community structure. And this will be done using 2 very large-scale studies. One is a continental scale based in North America, and the other is a smaller regional scale study that's developed along a natural aluminum and pH gradient in the southwestern United States. And then the third is actually, rather than looking at mere correlations, it's actually to look at aluminum and look at pH and to try to discern if there's either one or both are mechanism shaping communities. So we have to back up just for a second, and I know that everyone's really familiar with the relationship that's been proven over and over again between pH and community structure and diversity. So, but I'll go over it briefly in using this figure from Labor et al. 2009. And what's really nice about this is that they modeled a nice curvilinear relationship between pH and phylogenetic diversity. And you'll see that I've drawn this line here. This is just an arbitrary line somewhere between circumneutral pHs 6 and 7. But you'll see that at that point we see diversity saturating and then decreasing on either side of this pH range. And so the question is now is the mechanism, is it pH or is it not, or is it in part pH? And I think what we could do is, looking at this curve, is that for the sake of building a hypothesis, we could divide this up into discrete segments. So for example, you could draw a line through this segment. This is not a linear regression. This is just an arbitrary straight line through these points. In this section here, you have circumneutral pHs with the highest diversity and then decreasing diversity as pH decreases. And you could do the same thing on the other side of this curve. And then you could, if you wanted to, you could even put a straight line with no slope here where diversity saturates. So, but there's a potential that there's different mechanisms operating in each one of these sections of this curve, or one mechanism, series of mechanisms. So for the purpose of the talk today, I'm just going to focus on the pH range here from circumneutral pHs and decreasing to pH of 3. And then I'm going to propose that what's governing the trajectory of diversity here is actually aluminum 3+. And so I think that in Europe that there's more awareness of soluble aluminum and its effects on plant species, aquatic invertebrates, that perhaps there are, at least in southwestern United States and western United States. But just in general, I think that what I'd like to do is just review with you over this pH range what solution chemistry looks like in soil. So here we have a diagram that shows the relative abundances of different cations in solution at any given pH in a typical mineral soil. So again, I'm going to draw an arbitrary line here between 6 and 7. So it's not so arbitrary because in the previous diagram, I actually also drew a line at pH 6.5 where diversity saturates. So at this point in the soil, you see the soil solution dominated by calcium and then smaller amounts of magnesium and potassium and sodium, which are what we refer to oftentimes as the base cations, necessary critical nutrients in soil solution. And at the bottom, there's a tiny sliver there in green, and you trace over to the left-hand side, you see that represents hydrogen protons and an aluminum 3+. And so if you remember then that diversity saturates at this point, and then you move to the left on this as pH decreases, you see incremental decreases in the base cations and increases in the acidic cations, with a pretty precipitous increase here at pH 4.5. Okay, so why aluminum? So if you're not already aware of it, aluminum's toxic to most organisms. This is a diagram that shows the effects on root structure in a soil as aluminum increases. And the most toxic form that I'll be talking about today is aluminum 3+, although I will probably be dropping the 3+ for the sake of brevity and be referring to it simply as aluminum. So not only is it toxic to organisms, but that it also complexes with nutrients in soils, and it pulls them out of the bioavailable pool. And the two primary victims of this are low molecular weight organic carbon and phosphorus. And this is not, of course, aluminum is not a situation where you have discrete quantities and discrete locations. It's not like taking getting a bulldozer and then excavating out and removing it, because aluminum's everywhere in soil. It's a critical component of clay minerals, and it's the third most abundant element in soils. Okay, so the overarching hypothesis of the talk, at least for two of the studies, is that aluminum 3+ aluminum drives bacterial community structure in soils below circumneutral pHs. And I have a series of sub-hypotheses, and these dovetail with the patterns that we've already witnessed between pH and community structure. So just briefly, we anticipate seeing that with increasing aluminum, we'll see diversity decrease. We also expect to see strong correlations between aluminum and bacterial community structure. And then we also, in attempts to model systems, we anticipate seeing that aluminum emerges as an effective predictor. And then there's a whole suite of different hypotheses that we could make looking at effects on taxa abundance. And I'm not going to go through this list. It would be exhaustive. But basically, with increasing aluminum, you expect to see increasing numbers of acidobacteria or decreasing numbers of actinobacteria. Okay, so the first study across North America. Okay, we were really fortunate to be working with the FEAR lab. They were very generous to provide the pyrosequence data from the Lauber et al. 2009 paper, and then also provide us with chemistry data that included the pH and cation exchange capacity, base cations, base saturation, basically indicators of soil nutrient status. And then I was able to acquire the samples as well, the soil samples, and with the potassium extractable solution, I extracted soluble aluminum, which I used as a proxy for aluminum 3+. So here's what we found. Okay, so we found that diversity decreases with increasing aluminum, which is what we anticipated. We found a significant linear relationship over this pH range. And this is a linear regression through the filled-in circles. You'll notice that there are circles here that are empty circles. And we determined through successive analyses that these are actually outliers, at least for the sake of this hypothesis. They are aridosols. They are desert soils. They are basic soils, highly alkaline soils with a low precipitation, low moisture content. We believe that there are another hypothesis should be formulated to look at those particular patterns in those samples. Okay, so for the same samples, we developed molecular fingerprints, and this is, we use a weighted unifrac and NMS to develop this graphical representation of the community structure. And I also took each sample and I binned them according to the aluminum concentration. So if you look over here on the right-hand side, we have the pluses, samples with pluses representing them are the samples with the highest aluminum concentration. And then the infilled circles represent those samples with the lowest concentrations. And you notice that we have a lot of these circles that are filled in, and that is where diversity saturates on the previous diagrams that you saw. So we see this trend where the community is separating out on this continuum of aluminum concentrations. And then when you look at the univariate relationships between the soil chemistry and the community structure, you see that there's a very high correlation significant correlation between aluminum and community structure. So that circle that surrounds this diagram represents a Pearson correlation coefficient of 1. And the straight lines are the Pearson correlation coefficients. So for aluminum, that Pearson correlation coefficient is 0.94, which is very high. Now, on the other side, though, we have equally high correlations between the other soil chemistry that includes the base saturation, base cations, and pH, but that we don't see that relationship with sodium or cation exchange capacity. And I've also circled in those samples that I consider outliers, the iridosols, and I removed those from subsequent statistical analysis. Okay, so this next figure here is our attempt to model these particular samples. The idea is that we have all these correlations. We have all these univariate variables. We have all these correlations, but is there a particular predictor variable or series of predictor variables that emerges that could explain the distribution? We've already seen that pH is an effective predictor. Now, what I used was a modeling program and primer called DistalM, and I combined it with distance space redundancy analysis. And I also used Akaiki's information criterion to develop the models. And so it generates a series of models based on 1 variable, 2, 3, 4. And I was looking for the most parsimonious model. So one of the best models was actually a 1 variable model, and it was based on aluminum. And if you look here on the y-axis, you see the 34.1%. That means that with this model alone, I'm able to explain 34.1% of the variation in this spatial representation of the community structure. That's a third. And it's a straight line. So basically, it's just lining them up based on aluminum concentrations. You see, I'm kind of missing the actual breadth, the depth of the spatial distribution. So we passed on to 2 variable models. And what we found was that addition of base saturation helps us better approximate that spatial distribution. This particular model, by adding aluminum, adds about 3 to 4% in terms of explanation of the total variation. So not a lot, but it's interesting to see the changes when you add that particular variable. Now, the thing that's important to note is that I added base saturation. If you look at calcium or potassium or even pH, you can generate almost the same model. But you cannot, in a 2 variable model, you cannot duplicate this unless you have aluminum in it. So it's a critical part of this model. Okay, in terms of taxa, we see the similar patterns that you see with pH, so I'm not going to dwell on it. On the phylum level, the only phyla that shows a positive correlation with aluminum is Acidobacteria, and the other ones show the inverse relationship. Okay, so let's pass on to something that's a little bit more challenging. We developed a regional scale natural aluminum pH gradient, and the reason I say this was challenging was because our soil pedologist was working with all the soil horizons. We were staying in the upper, in the A horizon, and in our portion of the soil, the pH range was only about 1 unit. So what we were going to attempt to show along this 1 pH unit change, we wanted to see if the community structure would change. So that means if you look at this relative to the previous continental scale study, that it's that very small portion of the curve, which potentially is right where diversity saturates. And then when you look at the soil solution chemistry, we're looking at chemistry between those 2 red bars. So the very, very small changes in soil chemistry over that range. We had an array of chemistry data because we were working with soil scientists who like to analyze every soil parameter possible while they're determining soil taxonomy. And I'm only going to show you some of the parameters that were measured. We had the same data, types of data that we had from the continental scale study, although we had detailed information on soil mineralogy and on the forms of available iron from mineral phases. Okay, so just to give you an idea of how we generated this pH aluminum gradient, this is from a litho sequence. These are actually very rare in the literature because it's similar to a chrono sequence, except that you're not varying the time of soil formation. What you're going to vary is you're going to vary the bedrock, the parent material, and the parent material will shape the soil that develops from it. So we had parent material that served as reservoirs of varying aluminum. And so from the left-hand corner, we have the dolostone, which would represent the soil with the highest pH. And then we go in order of decreasing pH with the basalt, then the granite, the right-hand corner, and then the rhyolite. And the overstory was held constant. This was in a temperate Pinus ponderosa forest. Okay, so we found there was significant separation by site, by parent material. And now, because it was on a really, really narrow range there in terms of pH and aluminum concentrations, we found that the samples clustered. So on the right-hand side, we have the rhyolite and the granite soils here with the higher aluminum. And on the other side, we have the low aluminum, high pH sites clustered together. So we wanted to model this just like we modeled the continental scale data. And this one was just kind of interesting because what we found was, yes, we had to incorporate aluminum in the models. So if you look here on the axis 1, we have 36.6% of the variation explained by this model. So the straight line is extending out to the aluminum. You can see where they're separating out the low aluminum sites from the high aluminum sites on the right. They're separated out along this axis here by virtue of the aluminum content. But then on the second axis, which explains 11.4% of the variation, we see that it's the iron mineral phases that are important in separating out those communities. Okay, so in terms of taxonomy, what did we see? Well, so I know that we're oftentimes resting here at the phylum level, but I think that it's masking some of the critical responses. So if you go to the level of order, in this instance, we had about 50% of the community at the order level that was tracking one of two distinct chemical profiles. And so I've color-coded this. This is a table showing the correlation coefficients between the orders and the particular chemical parameter. So there you have your forms of available aluminum, and there's your available iron, and then there's your pH. So the two chemical profiles are, you have high bioavailable aluminum, you have low pH, low amounts of the bioavailable iron. And then the pool, the second pool is the inverse of that. You have low bioavailable aluminum. Okay. So then, okay, we have all these correlations. Correlation, correlation, correlation. They're just, the literature's wrought with them. I've seen a lot of them today. We don't know. Is pH the mechanism? Does it matter? I think that it does matter. But we wanted to evaluate whether aluminum and pH or aluminum or pH are the actual mechanisms shaping communities. Well, it's difficult to do this, to tease this apart, especially when you're working with survey-based data. And so what we did was we looked at this pH aluminum gradient as an opportunity. So the rationale is this. You have acidic soils, and the rationale is that if you have acidic soils, they should harbor acidophilic organisms. It should be more abundant than if you were looking at a soil that's less acidic, higher pH. So we anticipated that if we use a selected media that would target acidophiles, that we'd have a greater abundance of acidophiles in the low pH soils as opposed to the high pH soils. So there's our hypothesis. And then we also developed the same hypothesis with aluminum. So if you are looking at soils with high reservoir, serving as a high reservoir for toxic aluminum, you would expect to see greater abundance of aluminum-tolerant organisms. So we had two treatments, one which is a low nutrient broth with pH of 4, and then the other was the same conditions but with 100 millimolar aluminum addition. Okay, so very, very, very simple. Results were not what we anticipated, okay? So this diagram shows the results from the culture. We have parent material on the bottom axis, and on the y-axis we have the cell densities, the abundance of the acidophiles. And then I show here what the pH conditions were across the sites. Well, there's no trend whatsoever with the culturable acidophilic heterotrophs and the hydrogen proton loads that are present in the soil, and that was unexpected for us. So conversely, though, with the aluminum additions, we saw a predictable pattern. We saw that the soils that have higher amounts of aluminum, that's because of the parent material underneath, they have higher abundance of aluminum-tolerant heterotrophs. Okay, so where does that go? Where are we right now? Okay, like I said, we've got lots of correlations, and we've got a very simple culture-based manipulation. So first, I think, though, that I think it's worthwhile to open up the discussion to aluminum as actually being a viable driver. We see very good relationships between diversity and community structure at multiple scales, relationships with taxa. The models are requiring aluminum. They are better than if you just use pH alone. And we're able to explain up to 35 to 50% of the variation at these scales. I think that's phenomenal if you to think about. Of course, it still means we have either 50 to 70% still to figure out, so we've got our work cut out for us. Along the gradient, I think the gradient was really telling. So we're looking at a very narrow pH range and a very slight differences in chemistry. And so the fact that we saw difference in the community structure along this site shows that communities are very sensitive to the chemistry. And that parent material also open for discussion as well. That parent material is important for shaping communities. And then, of course, we didn't address anything in terms of function, so that's a wide open question as well. Okay, so mechanisms governing abundance on the gradient, pH versus aluminum. Well, there's, at least over this narrow pH range, there was no relationship between the acidophile abundance and the hydrogen proton loads. Okay, but we're just talking about this narrow range between 5.5 and 6.5. But we do see a predictable pattern between the aluminum chemistry in the soils and aluminum-tolerant organisms. So where to from here? So the question is, we really need to assess aluminum versus hydrogen protons over a wider pH range. Okay? And then it would be potentially useful in going and reevaluating some of our older data sets in the context of aluminum. I think that this actually is important because I think it could actually change management practices. Rather than managing soils for pH, we'd manage them for aluminum, and I think we'd come up with some very different strategies. In fact, some of them are already in operation. Ian Saunders' lab works with AMF and actually has a program on the ground running in Venezuela where he's using AMF that shows high tolerance for aluminum, and he's pairing up with cropland plants. So it's a very different strategy than saying, okay, well, we're just going to go find some calcium carbonate limestone and sow the field with calcium carbonate. So these might not be options that are available, especially in tropical and subtropical countries. So I'd like to acknowledge my collaborators and my funding sources, and I thank you for allowing me to be here today.
Thank you very much. Okay, so we have some questions. Not so many, everybody, yeah.
Thanks for this interesting hypothesis. I just want to know if you, based on this aluminum toxicity, you will think that tropical soil have a lower diversity than temperate soil?
So you said in terms of tropical soil versus a temperate? What was your question?
Yes, versus temperate soil. Tropical soil versus temperate soil will have a lower diversity because you have more aluminum and more acidic.
I'm having a hard time hearing that.
Okay.
Oh, in the tropics? In terms of lower diversity
Yeah.
in the tropics?
Yes.
Those soils are, yes, well, they're adapted as well, but yes, in general, the diversity is, oh, sorry, that the diversity could be attributed to aluminum toxicity.
Okay, it's a very clear result. I'm very curious about the effect of plants that might change the chemistry of aluminum in the rhizosphere.
Well, it's very well documented, the aluminum toxicity, the effects on plant communities. And so that there would be definitely interactions there that would affect the communities as well. Yes, most definitely.
So it's annoying to work with soils. When you change pH, you also change the aluminum availability.
Yes.
But do you think that you can compare this data set with an aquatic one, where you wouldn't necessarily predict that you have the same association between pH and aluminum, or at least you could try to factorially separate between them, experimentally?
Yes, it would be easier because they're less complex. And yes, and I think that there's been some work already been done in aquatic systems, at least with the eukaryotes like algae and so forth. But that would obviously be an easier place where you could actually test this hypothesis, where you could actually decouple the two. But it's difficult. Yeah, they co-vary. They always will.
Yeah, I'm just wondering, pH kind of works pretty well across very broad soil gradients, but I've seen if you cut up your gradient like you did, then you'll get different drivers within each.
Yes.
So if would aluminium, does it about 6.5, it's
Yes.
not non-existent over that, so as an indicator, maybe you'd start looking at calcium. And
yeah, so it's all completely driven by solubility dynamics. Yeah. So where you see the diversity, you see a really sharp decline, and then you hit almost like a sort of a threshold, like I said, where diversity saturates. It's exactly where you see aluminum start being in measurable quantities.
Okay. Did you have any? Do you know if there was any calcareous soils in that in the 90 in the survey?
Yes,
Yeah.
and then in fact the iridosols.
Right.
And so I think so if, for example, if you were going to select site. And you had soils above 6, I really wouldn't worry about the aluminum aspect of it. But if you're going to go a range of soils, circumneutral, and drop it below 6, I think that maybe it might be worthwhile be measuring aluminum. Yeah. And you go on the other side of the curve, you're looking at, I think, another mechanism. I speculate you're looking at salt concentration. Salt, either you're looking at sodium or calcium.
Calcium,
Yes.
I've seen that above.
Yes,
Yeah.
and that's what I think that we need to do on the other side. Now, that big lump in the middle is a scary place, because that's not that simple, and that's where, I mean, that's where it's abiotic, it's complicated.